Okay, well, good morning, everyone. It is Thursday, and it is the last day of the week for me, anyway. Probably for most of you as well. Who has lab tomorrow? Okay, so some of you have lab tomorrow. Remember that at this point, you have all the information in your hand for the first exam. Okay, so we finished up Chapter 2 last time, and the first exam, which will be next Thursday, is going to cover Chapters 1 and 2. Chapter 1 was the introduction stuff, the atom to atom, the body systems, the hierarchy. Chapter 2 was all things related to chemistry. And then it's also going to include the information that you learned in Lab 1. Directional terms, body cavities, serous membranes, directional terms, all that good stuff. And some of you have already been quizzed on that yesterday in Lab. Some of you are going to be quizzed on that tomorrow, and some of you will be quizzed on that on Monday. But regardless, all of you will have studied for and been quizzed over your first lab materials, and all of you will also have done lab number 2 before that exam. And that's why the exam's a little bit later, like I think I've mentioned, than it would normally have been. So because of the Martin Luther King blip in the schedule. So are there any questions about how to proceed in preparing for the first exam? You also have vocab 1 through 16. Yes, ma'am. Let me take a look at that in a minute, okay? Okay. Anybody else having issue with Chapter 2?
I did, but I had to call back. He said I had no longer had access.
Interesting. Okay, let me take a look at that. But you shouldn't have to do that. So let me take a look and see if there's something on my settings. What did they tell you? It's something about...
It said that I had access until my class was expired.
Interesting. Okay, let me look at that. I got one email and I couldn't quite figure out what it was saying, and now that's the second confirmation. There's something in there. I'll look at it. I'll look at that as soon as I get done with this, and so that should be on.
The due dates for chapter 2.
Okay, let's talk about the due date. So on the calendar, on the mastering calendar, keep in mind that this is the same mastering calendar that's used for you guys who have lectures on Tuesday and Thursday, and those who have Monday, Wednesday lectures. There's a couple things you need to do. There's the study modules, and then there's a homework assignment. The homework assignment says on the schedule that it's due a week from today, right? It says the 27th. And the reason it's on, is that the right 27th? 29th? The reason it says that date, that's the last possible opportunity for anyone to do that homework. Okay, that's a deadline. But ideally, you should have already started, you should already have Chapter 1 homework done, I would hope. You've already looked at it, right? And you should be on your way to doing Chapter 2 homework. You definitely want to do the homework assignments. They say Chapter 1 homework or homework Chapter 1. You definitely want to do those assignments before you do the quiz. Now, the quiz is set up to be due no later than next Tuesday night at midnight. Okay? And that quiz is in preparation for the exam. So I would definitely do the homework assignments before you tackle the quiz. The quiz is open book. It is open resource. It is untimed. But it has a deadline of midnight, so I would not start it at 11 o'clock, right, on Tuesday night. I would go ahead and just take a look at it. You can go in and out of it as many times as you need to and do really well on that quiz. It will help you. You won't see your answers until after everyone has taken the quiz on Tuesday at midnight. Okay, so once the Tuesday midnight hour comes and everyone has taken it or not taken it, at that point, it will open up and become available for you to review your answers. Okay, and that's why it's a couple days before the exam, so there is time for review after the quiz has been completed.
There's an exam that's due today at midnight.
Not an exam.
Well, it says exam 1 or something like
It says there should be exam 1 quiz, and that's due next Tuesday.
that. Oh, on the calendar it says it's due today.
Not my calendar. Show me afterwards, okay?
Yeah, I don't have a quiz at all showing up.
The quiz became due or became available, I think, 2 days ago. So it should have popped onto your calendar as of either last night, midnight, a day and a half ago. Okay, it came online. So if you haven't looked at it in a couple of days, take a look at it. Some of those things don't appear until they're more timely. Okay, so let me know if there's any email concerns. Let me know by the end of the day so that we can get these things resolved. But I'll be in my office this afternoon. If you have any questions, actually, I'll be in my office through 1, and then I'll be in my it'll be in the lab. You can always find me in the hallway, okay, in the lab, if you have a question for me. Okay, and then I have a lecture this evening. So I'm around till at least 6 o'clock. So email me if there's any concerns. Let's get these things taken care of before the weekend comes, definitely. Okay, any other review content concerns from chapters 1 or 2? The other thing you want to do, if you haven't already, is to look at your pre-lab number 2. And in that pre-lab, there's a discussion, some questions about ionic bonds and covalent bonds and water and sodium and chloride. And that will, I think, if you're fuzzy at all on this conversation, I think it will help solidify some things. And then Martini has a wonderful, description in the first couple modules of chapter 2. And don't forget, there's also practice quizzes and activities on your mastering site as well. So lots and lots of practice, and hopefully by the time the exam comes around next week, you're feeling pretty comfortable. The exam's going to cover 1 through 16 vocab, and I think I owe you the last 4 terms. So let's take a look at those terms, starting with 13. And I am recording today's lecture. I wasn't happy with the recording I had of chapter 3, so I'm recording this again, and I'll post this as a fresh recording. But 13, 14, 15, and 16, these will be the last ones. Now, the vocab, there's probably 65 or 70 or so terms that are on these first 16 slides. Of those 60-some terms, I'm going to choose 12, 13 of them for the exam, okay? So it's not like you have to know every single one of them, but this is the collection of terms from which I will take my questions. So, capito, off with their head, if you will, capital punishment. Capito means head. Carcin, cancer, carcinogenic, something that causes cancer. Cardio, we know that one, the heart. And carpo, or carp, the wrist. The carpals are the wrist bones. The carpus is a directional or a body area referring to the wrist. Cat, you've seen this term. Catabolism was the breakdown, reactions that break things down. So cat means breakdown. You've seen caudal in the lab, that means toward the tail. Cephal, toward the head, so cephalic as an example. Centesis, we've all heard of amniocentesis. It's a surgical puncture to remove fluid. Specifically in amniocentesis, we're taking it from the amniotic sac. There's a fluid sac around the baby, but it's the removal of fluid through a surgical puncture. And then cerebro, the brain. Cerv, the cervical region is the neck. Chondro, you've seen this term as well, hypochondriac, right? That region up underneath the ribs, the uppermost lateral sections of the abdominal pelvic cavity. Chloroplasts, anything related to green, okay? So chloro this, chloro that. And then chrono, is time. If you have a chronic condition, you are going to have it for a long time. Chrono. Chromo, color. You guys are probably all way too young to remember film for cameras, but there was Fujichrome and there was Kodachrome, and this was the colored film for cameras. And so that's the way I remember it. Chromocyte would be a cell that gives off some sort of color. Chyme, juice, in your digestive system. Once the food goes to the stomach, it leaves the stomach as kind of a juicy, pasty-like substance called chyme. Side, to kill, spermicide, homicide. Circum, around, circumnavigate, circumduction, something going around. And finally, cero, meaning yellow. If you have cirrhosis of the liver, one of the symptoms of that is a yellowing of the eyes. And so the cero refers to yellow in color. So those are the terms, right, for the first exam. I think I saw a show of hands last time. Most of you had made up your flashcards and are moving right along with that. Please never feel obligated to wait for me to go over these, but I do want to review these with you each and every lecture. Keep in mind that that's the only thing that's on the exam right next week that I just went over on vocab. The rest of this presentation over Chapter 3 will not appear until the next exam. Although I think sometimes even going through this Chapter 3 material, there may be some connections that you make that may help you better understand Chapters 1 and 2. Anything at all? Exam next week. I'm still going to see you next Tuesday, right? And we're still going to continue on with Chapter 3. So I'm not done seeing you before the exam. But remember, you do want to have an 884-E form, a Scantron form. That's a full-size green form. Pencil with you next Thursday. That's all we'll do. It's just the exam. It'll be about 100 questions. It'll be largely multiple choice. About 85, 90 of those questions will be multiple choice. There will be a couple fill-in-the-blank questions. The fill-in-the-blank questions will be mostly the vocab. Okay, so the vocab will be fill in the blank, and there will be a couple of images to know from the screen. Definitely be able to recognize your body systems when you see them. Definitely know your directional terms and that sort of thing. And don't forget to review that Darley article, that first page that was in your supplement, two-sided page about study habits and how to be successful. Definitely, if you haven't read through that, do that. And that'll be the exam, okay? It's the shortest exam of the semester. It's about 100 questions. Some of our exams as we go through will be closer to 150 questions. So this is a nice, gentle ramp up to what to expect for the rest of the course. Okay, so this is Chapter 3. And Chapter 3 is about cells and organelles. And you recognize we're on that continuum, right? Atom to atom continuum. So last time, Chapter 2, we talked about atoms and molecules and macromolecules. And in this chapter, we'll be talking about organelles and cells. So that's our focus here. We'll also be talking about tissues next time. And then in about 10 days or so, we'll start talking about our first system. And that'll be the integumentary system. But that's not too far away. Here's a review question. That's good. What section or plane is used to create these figures? How about letter A? Take a look at that. What section or plane was used to create that? I'm hearing sagittal. That's absolutely right. You could also have put mid-sagittal. And letter B? Frontal, also called? Coronal. Make sure you know both terms. They're equivalent. And finally, C? Transverse or cross-section. I heard them both. Absolutely. So transverse or cross-section. Make sure you know the equal terms. Okay, so some of you may have recognized when I introduced myself that my background is in cell biology. My undergraduate, my master's degree is in cell biology and microbiology. I really like this stuff, but I promise you I won't overwhelm you with details that you don't need. But I think maybe you'll find this really, really interesting. Cells are the, as you know, the functional smallest living thing when we go through our atom-to-atom continuum. And it turns out that these are the building blocks for your body. Your body has somewhere around 75 trillion cells. It's an amazing number, not to memorize, but just an awesome number of cells that are making up your body. And you are making, to make it even more awesome, about 25 million cells every second. Right? We are making 25 million new cells in our body every second, and that means we're also destroying about 25 million cells every second, just to keep our homeostasis. So pretty awesome processes. There's about 220 or so different types of cells in the body. Each cell has a slightly different makeup, does something a little bit different. And there are even some cells in the body that we're still not quite sure what their function is. We know they're different than the other cells, but we haven't quite figured out what their primary purpose in life is. So there's still a lot of active research in cell biology. I described to you in the first lecture the characteristics of life. We talked about how that's true of not only the organism, you and me, but also of individual cells. This is the same list, worded slightly different. So that means that cells, right, to sustain life, must obtain their nutrients and get rid of their waste products. That's the idea of metabolism and the idea of taking in energy and using it. They're going to be made of smaller units. That's going to give them a shape, a certain shape. We're going to see cells come in all sorts of shapes and sizes. Cells are going to reproduce. They're going to divide and make more of themselves. That's true of most of the cells in our body. Can you see an individual cell? Can you see with your naked eye an individual cell? No would be the best answer, but yes, there's one cell. There's one cell that you would be able to see with the naked eye. It would be microscopic, the little tiny end of a pin, and that's the human egg. Okay, so the human egg would be the biggest, roundest, most easy to see cell in the body. Really, anything else would require a light microscope. Now, on the screen here, everything on the top is what can be seen with the unaided eye, right? Unaided eye. And you see it goes down to about the level of the human egg. Above that, it does say muscle and nerve cells. It's a little bit misleading. Skeletal muscle cells, and we'll talk about them in a couple of weeks, but skeletal muscle cells are as long as the muscle is. So you have a muscle in your leg called the sartorius. It kind of wraps around your thigh and goes around. It's a really long muscle. The cells in that muscle are as long as the muscle is. And you have neurons that are traveling from your spinal cord all the way down to your big toe, those can also be 3 feet long. But they're skinny, skinny, skinny. You would not be able to see it individually. Okay? They are very, very long, but truly they'd be so thin like you wouldn't be able to see an individual cell. The human egg is round enough that you would actually be able to see it with the naked eye. Then we see the things in the yellow area, light microscope, that's what we're using in the lab. And here we see cells. Right, so we'll be looking at a lot of cells. You, in lab this week or next Monday, will look at cells undergoing mitosis. In lab 3, we'll be looking at about 12 different tissue types under the microscope, and we'll see cells there as well. Also with the light microscope, you would see smaller cells like red blood cells. Again, in lab this week, you're going to get a glimpse, not under the microscope, but on the screen, of what red blood cells look like. And you would be able to see larger organelles like bacteria. That's what you're going to be able to see with the light microscope. If you want to see something smaller, you're going to have to use an electron microscope. We don't have those hanging around. Those are pretty specialized. And with with an electron microscope, you'd be able to see viruses and see small organelles like ribosomes, or even large proteins like proteins and lipids. Excuse me. It suggests on here that we can see atoms with an electron microscope. The last time I looked, and I have not seen a new report, I don't know that we've ever actually seen an individual atom with an electron microscope. I know that we've seen evidence of atoms, but I don't know that we've ever actually seen, that is an atom. We've seen, we talked about atoms, right? Protons, neutrons, electrons. We've seen the evidence of atoms, just like we see the evidence of the wind, but I don't know of any conclusive report that says, that's an image of an individual atom. So this is an electron micrograph. Usually light micrographs, things you see under the light microscope, are colored. You'll see dyes, and light is important. In electron micrographs, usually they're black and white. There are color-enhanced ones, and I actually have one later in the presentation. But typically, when you see in a textbook a black and white image of a micrograph, it's typically taken with an electron micrograph. You'll also see in the bottom of most images a little scale. So it gives you an idea of the scale that you're using to appreciate the size. Don't memorize the numbers, but it tells us that cells are pretty small, from up to 140 micrometers in diameter. That would be the oocyte, and again, that's the only cell that you'd be able to see with your naked eye. And to see smaller things, as in this image, you would have to use an electron micrograph or microscope. Okay, so what are cells doing for us? I think we have a decent appreciation of this, and this is just a short list of some of the things that cells do for us within our overall body. They're definitely covering us, right? Our skin, the outer layer, is made up of epithelial cells. In lab 1, we talked about serous membranes, talked about the visceral and the parietal layers. Those layers are epithelial cells. They're covering and coating and lining. All of our blood vessels are covered by cells. Movement, right? The ability of your muscles to move is the result of proteins and the cells moving within those cells. Ligaments and tendons, things that connect us, they are fundamentally made up of cells. We have white blood cells, leukocytes, that are a very big part of our immune system. We have nerve cells, neurons, that allow us to communicate rapidly from the brain to the rest of our body. And finally, we have reproductive cells, the egg and the sperm. So we see that cells play a varied role within our overall body. There's really a couple of big objectives for this chapter, what you need to be able to walk away with knowing. One is to label the cell. You did some labeling of the cell in your pre-lab number 2. We didn't talk too much about the cell in lab number 2, but you did do some pre-lab labeling of it. So you're not completely unfamiliar with something like this. So you need to be able to label the organelles of the cell, and then you need to be able to tell me what each organelle does. Right? What's the primary job, purpose of the organelles? That's really what the first part of this chapter is all about, and what this presentation today will be about. This is the prototypic cell. This cell would never exist in your body. This is an artist's rendition of a cell. And I know this cell would never exist in your body because there are some things on this cell that would never coexist. For example, you would never have both cilia and microvilli on the same cell. Those would never coexist. And so this cell is definitely an artist's makeup just to show us different ideas. And different cells in your body would have slightly different arrangements of these organelles. For example, a liver cell, yes, it would have all of these pieces and parts, most of them, but it would be quite different from a kidney cell or a brain cell or a skin cell. So we'll see there's some variation in these organelles, but this is that prototypic cell. Let me just kind of get us started with this, and then as I move through, we'll spend some time talking about the plasma membrane, and then we'll finish up this morning talking about a handful of the different organelles. We won't quite finish this presentation, but we'll get toward the end. Do not memorize colors on these organelles. Different artists will use different colors, so don't get caught up on color. Instead, look at the arrangement. Look at the relative location of these organelles within the cell. So we'll start in the very center. So we got this big old purple nucleus, right? I'm saying purple because that's what it is, right? But big old purple nucleus, and that's where the DNA is. Now that's a different word, a different meaning than nucleus last week. Right? Last week we talked about a nucleus. The nucleus was the very center of an atom where you found protons and neutrons. This is a cellular nucleus, which is the command center of the cell. And we'll use the word nucleus again when we get to the brain in a totally different way. So the word nucleus has multiple meanings within our field. In the very center of the nucleus, I'm able to see a dark staining little ball. That's the nucleolus, okay, or nucleolus. You'll hear it pronounced both ways. We'll talk about each of these in a few minutes. Around the nucleus, there will be a series of membranes, cell membranes, and that's called the endoplasmic reticulum. Now, endo means within, plasma membranes, reticulum means a network. So it's a network of membranes within the cell. Okay, that's really what that name means. And I don't want you to get caught up that it's just rounded in two dimensions, if you will, but truly the nucleus is a sphere, and the endoplasmic reticulum goes around the, around the nucleus in all dimensions. Okay, as a sphere kind of structure. This is just a cutaway. There's two flavors of endoplasmic reticulum. The inner layer typically is studded, and you'll see little dots on here, and those little studded dots are ribosomes. And then there is some of the endoplasmic reticulum, usually a little bit further away from the nucleus, and I'll draw it out here. This series of ER has no ribosomes associated with it. That is called the smooth ER. Because it appears smooth, it doesn't have the ribosomes attached to it. Further away, even moving away from the nucleus further, there will be a series of folded membranes. Looks like that Christmas ribbon candy, if you've seen that in a few years. And that is the Golgi apparatus, or just the Golgi. All of those membranes are continuous. They all are connected one to another, from the nucleus through the ER, through the Golgi. It's really one big bleb of membranes with slightly different jobs. Within the cell, there would be a lot of these big bean-shaped structures. Those are mitochondria. There would be a pair of these centrioles. Looks like little Lincoln logs, some sort of little tinker toy for building. Those will be really, really important in cell division and in mitosis. Each of those little centrioles will go to the north and south pole and will spit out those mitotic spindles and pull the chromosomes back during mitosis and cell division. Inside the cell, I see some little vesicles, little vesicles. They will come in different flavors. There'll be lysosomes and peroxisomes, and we'll hopefully get to that conversation today. And then also within the cell, I'm seeing lots of strings. All those strings are proteins, and they would be collectively referred to as the cytoskeletal proteins. Just like your body has a skeleton of bone that gives you structure, the cell has proteins inside that give cells their unique shapes. Some cells are very long and skinny. Some cells are very big and round. What makes cells different is their cytoskeleton that gives them shape. On the outside, we've got the cell membrane. And then there are some different processes or things that stick out. In anatomy and in biology, a process is something that sticks out. So there are cilia, and in other cells, there are little tiny guys called microvilli. The only process that this picture does not show, but one coming up does, is the artist could have shown a long flagellum. Now, the flagellum is only seen on the sperm, but regardless, it's another process that we'll see as we talk about cells in this chapter. So those are the pieces and parts. right? Labeling them, and then as we go through, what do each of these different organelles do for the overall well-being of the cell. We'll just say a couple things about the nucleus, and that is, it's the control center of the cell. It is going to, inside, have most of the cell's DNA. It's where the chromosomes are going to hang out. And it is going to direct, really, all of the processes of the cell. This is a cartoon representation of a nucleus. It looks at first glance that maybe this is a cell, right? This may be just a whole cell, and this thing in the center would be the the thing in the center might be just the nucleus. But I want you to see something. Look at this membrane around the outside of this nucleus. It is not continuous. It's a little bit weird. It's a double membrane. So the nucleus has a double membrane. It's sometimes called the nuclear envelope, okay, the nuclear envelope, or the nuclear membrane. In addition to it being a double membrane, it also has spaces in it. You see gaps in it, and those are nuclear pores. And so you would not see that in a normal cell membrane, right? This is a specialized membrane around the nucleus. And we'll talk later about what is it, why do we need to have those openings? What is it that goes in and out of the nucleus that is necessitating those big openings? That means that this dark center here in the cell is the nucleolus. And as I go through this, I want you to grab what each of these things is doing. Right now, just write down that it's the site, or circle, that it's the site of rRNA, ribosomal RNA, is made in the nucleolus. That word doesn't make sense to you yet, but it will. If you just want to put next to that, responsible for making parts of ribosomes, that would be fine as well. So the ribosome synthesis begins and is critical from the nucleolus. Now, everything outside of the nucleus that is inside the cell itself collectively is called the cytoplasm. Okay, the cytoplasm. And that's everything. That's the organelles and the fluid and everything. The big soup in there is called the cytoplasm. Specifically, the fluid component of the cytoplasm has another name, and that is the cytosol. So the cytosol is just the liquid, right? Just the fluid. And then when you add all the organelles to the cytosol, we call that collective alphabet soup, we call that the cytoplasm. Okay, just to clarify those two words, cytoplasm, cytosol. Here's just a quick cartoon of some of the organelles we'll be taking a look at. We've already seen the upper left. That is, we've already seen the nucleus. These are not to scale. Then the flattened set of membranes is the Golgi. And that other set of membranes that comes in two types near the nucleus, the rough ER and the smooth ER. There are mitochondria, little bean-like shaped structures that we'll talk about probably next time. There are little vesicles in the cell filled with proteins and other products. We'll talk about those. And then finally, this is one of those little centrioles that'll be important in cell division. And the centrioles themselves are made of microtubules. We'll come back to that. But the centrioles themselves are made of microtubules. So let's start going through these. And I'm going to spend the most time of any organelle on the plasma membrane. In lab this week, that's exactly where we'll start. And while we're going to be playing with dyes and auger plates and doing some other little demonstrations in lab this week, the primary thing for me to help you learn is imagine you're playing with a cell. And you've got a cell, it has a plasma membrane around it, and you know that every cell in your body has to get nutrients, right? Every one of those 75 trillion cells needs to have oxygen, needs to have glucose, needs to respond to other chemical signals in the environment. And every one of those cells is also making waste products and having to get rid of those waste products. There's a lot going on across the cell membrane. So we're going to spend most of our time here describing some of the processes where things go in and out of the cell. Now here's another artist's rendition of a cell. And again, the colors are slightly different, but you still see the same arrangement of organelles. You see the nucleus with the dark nucleolus in the center. You see the endoplasmic reticulum wrapped around it in two types, with and without ribosomes. You can see the Golgi. The artist used green here again, the green Golgi. You can see the mitochondria. You can see the centrioles that are going to be part of cell division. This artist did include the flagellum, okay, as one, right, a single flagellum. But there's also cilia shown here and microvilli. Again, they would never all coexist on a cell. So same pieces and parts. And as you label and look at different pictures, this is even a different picture than what you're going to have in your Martini book. You'll appreciate the overall architecture of the cell. Now, not all cells look like this, but we're going to start here. So the plasma membrane, really, really important. It is going to be the gatekeeper. It's going to make sure that only certain things can get into and out of the cell. It's going to be a barrier. It's going to prevent certain molecules from getting into and out of the cell. And like I said, every cell has molecules it must receive and other molecules that it must get rid of. We've got to regulate this. We've got to control this. So here is a simplistic cartoon representation of the plasma membrane. You've seen this molecule before. The reason I described phospholipids back in Chapter 2 was to prepare us to better understand this image in the cell membrane. So each of these little gray circled heads, right, that's that little circle head with the two squigglies. That is one phospholipid molecule. And what we see is that the membrane is full of these phospholipid molecules as a bilayer, what we call a bilayer, a double layer. And remember what was special about phospholipids? The gray part, the round part, represents the phosphate group, phospho. And remember that the phosphate group had a negative charge. If it has a negative charge, another way of describing that is that it is polar. Okay, so the outside of the membrane is polar, and the inside of the membrane is polar. Now, if something is polar, what does it tell us about what it can interact with? Like dissolves like. So polar environments can easily interact with water. Remember, water is a polar molecule, and polar molecules will interact with other polar molecules. So the inside of the membrane and the outside of the membrane are very polar. Another way of saying that is that the inside and the outside are hydrophilic. They have an affinity for water, right? Hydro, water, philic, a love of or affinity for. So the inside and the outside of the cell membrane is hydrophilic, aka polar. The inside, that double layer on the inside of the cell membrane, has a very different environment. Those little squiggly tails, remember, those were lipids. And remember that those lipids are by nature, by definition, non-polar. So inside this membrane, in this squiggly line area, inside the double layer, you have a non-polar environment. And another way of describing a non-polar environment is to say that it is hydrophobic. It is going to move away from water. Remember, lipids don't mix with water. So that's a hydrophobic or a non-polar middle layer. So that's kind of what you've got. You've got this bubble, basically, around every one of your cells, made up of a double layer of these phospholipids, which themselves create a little space, right, between the watery cell and the watery environment. In the membrane, there will also be a series of proteins. That's what this big green toilet paper roll looking thing is, right, in the cartoon. And these are membrane channels. You'll see membrane pore described as well. An opening that allows certain molecules to go into and out of the cell. These are not like big gaping holes where anything can go in and out of the cell. As you'll see in a moment, these proteins are very specific. They're only going to allow certain molecules in and certain molecules out. There wouldn't just be one or two types of these proteins in the membrane. There'd be hundreds, thousands of different membranes, proteins, each of which only allows a limited number of certain molecules in and out. Highly, highly specific. So that's our basic idea of phospholipids, bilayer with proteins sticking through the membrane. Now, if I went to Walmart and I bought some phospholipids, and I dropped those phospholipids into a beaker of water, they would kind of do what this is demonstrating. They wouldn't necessarily make a bilayer like a cell, but they would congregate. And let's make sure we appreciate what's going on here. These orange circles represent what? The phospho part, right? The polar part. And the polar part would be out here facing the polar water. And all those little hydrophobic tails would all gather toward the center, right? And move away from the water, hydrophobic, nonpolar. Okay, so that's what these phospholipids would do if you just dropped them in water, and we call that little circular arrangement a micelle, just as an aside. Yes? Okay, let's go back to that for a moment because it's going to be really important. So remember in chapter 2, when I introduced lipids, I said there were different kinds of lipids, and I said there was triglycerides, oils, fats, saturated, unsaturated. I mentioned steroids as being a lipid, and then I had a slide or two on phospholipids. And a phospholipid, recall in all the cartoons, was a circular head and two squiggles, right? The head, turns out, has a phosphate group in it, phospho, and the phosphate group by its very chemical nature has a negative charge on it. The long tail is what we called a hydrocarbon or a hydrophobic tail. Okay, so this is hydrophobic tail. So that means that the tail is hydrophobic, but the phosphate group is hydrophilic. Turn it another way to say that, the head is polar and the tail is nonpolar. Okay, so those terms are kind of interchangeable. Polar, nonpolar, hydrophilic, hydrophobic. Okay, they're interchangeable terms. So the cell membrane, what's in there? I've already kind of said it, but here it is in words. There is lipids, right? And those lipids are phospholipids. And then there's also a fair amount of cholesterol. Now, cholesterol, right, we know we need to minimize our cholesterol and keep our cholesterol levels in check. Our body makes cholesterol in addition to the cholesterol we take in our diet. But cholesterol is a very important component of your cell membranes. Without enough cholesterol, your cell membranes become a little bit too loose, a little bit too liquidy. So cholesterol is necessary to keep integrity, keep strength in your cell membranes. And I'll show you a picture in a moment where you can actually see the artist has drawn in cholesterol molecules to give the membrane some strength. Also, in the membrane, there will be some glycolipids. Now, whenever you see these words, I know they kind of start mixing together, but it is a lipid, right? That's the root is a lipid, and it has attached to it some sugar groups, glycolipids, lipids with a sugar group attached. And you'll appreciate these in the cartoon on the next page. In addition to the lipids in the membrane, there are also proteins. These proteins do all sorts of different jobs, but we're going to describe them as two different classes of proteins. Number one, those that are integral. That is, they are integrated completely within both layers of the membrane. So there are integral proteins. They are completely integrated. They pass through both sides of the phospholipid bilayer. So they peek out on the inside, they peek out on the outside. And then there are peripheral proteins, and as the name suggests, they're on one side or the other, right? They're on the edge. So they're either only on the inside or they're only on the outside of the cell, but they wouldn't be poking through both sides. And you'll see those in the picture coming up as well. And what are these proteins doing? All I've said right now is that some of them are integral and some of them are peripheral, but these proteins have some very, very important jobs within the cell, and here's a partial list of those. Number one, they transport things in and out, right? They allow certain molecules to move into and out of the cell like a tunnel, like a pore, like a channel. Other of the proteins in the membrane are going to be important for intercellular attachment. Inter as a prefix means between. So that means that between the cells, they're being connected one to another, right? Epithelial cells touch other epithelial cells and are attached to them. So something has to attach the cells one to another. The proteins in the membrane do that. There will also be anchorage for the cytoskeleton. The cytoskeleton were the proteins inside the cell, and some of those are going to help give the cell shape, Red blood cells have a different shape than epithelial cells, which have a different shape from neurons, which have a different shape from muscle cells. So that shape is in part from those cytoskeletal proteins binding to the inside of the membrane. Some of those proteins are going to be enzymes themselves. They are going to facilitate a chemical reaction. Some of those proteins are going to be very, very important in medications. For example, a liver cell has on its outside a slight different set of proteins than a kidney cell, than a brain cell. And that's what pharmacy companies do. When they're designing new drugs, you're going to take a medicine by mouth, right? It's going to go through all of your body. But that medicine's only going to target certain cells of your body. It's only going to target the liver. It's only going to target the kidney. Well, part of the way that the companies target those medications is specifically to these proteins found on certain cells, right? And so, again, there's cell-cell recognition. One cell looks different from another cell type because of some of these proteins. And we use it to our advantage when designing medications. And then finally, sometimes a molecule will touch the outside of a cell, but it will cause other signaling pathways to occur inside the cell. So sometimes a cell has a protein attached on the outside, which causes other changes on the inside. The molecule itself doesn't come in, but it affects what happens inside the cell. And that's called signal transduction. So there's a signal that's transduced or is sent across the membrane through these proteins. So these proteins are rich and varied and do amazing things within the membrane. And this image gives perhaps a little bit more of a complete picture. Still it's very, very simplified still, but it's more complete than the previous image. So in this image, I have my double layer. Here, the little phosphate heads are purple, and the little yellow tails, right, are the phospholipid tails between the membrane, double membrane. This is the inside of the cell. This is the cytoplasm, and this is the extracellular. This is the outside of the cell. The other name for extracellular fluid is the interstitial fluid. You'll see those words used interchangeably. Extracellular, outside of the cell, is also called the interstitium, or has interstitial fluid. In the membrane, I'm seeing big proteins. Here's a big blue one. I see it going through both sides of the membrane, so that's an integral protein. It appears to be one of those channel proteins. It's allowing molecules, you see the big hole in it, it's allowing molecules in and out. This green dude would also be an integral protein, right? It's going through both sides of the membrane. And then I see evidence of peripheral proteins. Here's one, this little yellow one, and this little yellow one. They're labeled as peripheral proteins because they're only visible on one side or the other. They're on the periphery of the membrane. Inside the cell, I'm seeing all these little proteins. Those are the cytoskeletal proteins. And inside the membrane, here's one, here's one, here's one. All those little yellow things with little rings in them, those are all cholesterol molecules. So there's a lot of cholesterol built into and creating structure within your cell membranes in addition to the other phospholipids and proteins. So that's kind of a more complete look at the membrane, but even this is quite simplified, okay? And then in this image, all these little green hexagons are sugar groups, okay? So there's a glycolipid. It's just showing you it is a lipid molecule with sugar groups hanging off from it. So there's just a lot of variety, lots and lots of variety on the proteins of the membrane. So as I've already said, the membrane is critical for allowing molecules in and distracting or not allowing other molecules in. And there's 4 different ways or so by which molecules get into and out of the cell. A, some molecules can go straight through the phospholipid bilayer. Some molecules are able to go right into the cell. Okay, and we'll describe those in a moment. If they do go straight in through the cell membrane, we're going to say that they are lipid soluble. In other words, they can dissolve in lipids. In lab 2, yesterday, tomorrow, and next Monday, you're going to be doing a little tiny thing with solubility. And you're going to be looking at how some molecules can dissolve in water and how some molecules can't. And how some molecules can instead dissolve in a nonpolar solution like acetone or paint thinner, and others can't. That little, simple little demonstration in lab should be going ding, 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 ding as I talk about this. Okay? okay, when you get there, or if you think about what you did yesterday in lab. So we've got some molecules that are lipid soluble. That means that they dissolve in lipids. What's another way to describe a lipid soluble molecule? Like dissolves like. Water is polar. Things that dissolve in water are also considered polar. So if something is going to dissolve in lipids, it would be a non-polar molecule, right? So lipids are non-polar, and they're going to dissolve in non-polar substances that we'll see in lab. Other molecules will get across the membrane through those channels, those pores that I've already shown you. Some molecules will have to be carried. They literally will have a helper molecule that will help carry them into the cell. And then lastly, there will be molecules moving in through vesicles, and I'll describe exo and endocytosis in a moment. So we've got 4+ ways by which molecules move into and out of the cell, and most of these are highly regulated. So we're going to spend a few minutes looking at this image. On the right, yellow, these yellow molecules are labeled for us. And it says that they are lipid soluble. So there we go. Lipid soluble molecules. And this picture suggests to us that they can just squiggle right through the membrane. These molecules are going from an area of higher concentration, look, there's 5 of them out here, to an area inside the cell where there's only 4. What is that called, those who've been in lab? Things are going from an area of higher concentration to an area of lower concentration. Diffusion, right? Diffusion. And diffusion is a type of passive transport. It just happens without energy. And we see these molecules are going straight through. And in lab, we'll also use the word concentration gradient. So the concentration gradient molecules are moving in. They're going down their concentration gradient. So we see that some molecules are lipid soluble. Put another word next to that. If they're lipid soluble, they are non-polar. What kind of environment do I have here inside the membrane? Non-polar. So these molecules are welcomed straight in and can pass right through the membrane. Have you heard it said in some commercials, women who are pregnant or think they are pregnant should not handle this drug? Does any commercial name come to mind? The one that's coming to mind for me is Propecia. And Propecia is a male pattern baldness medication that guys take. It is a steroid. What do we know about steroids? Steroids, cholesterol-based lipids. They would be able to go straight through the skin. This is why guys are wiping testosterone under their armpits, right? Low T. Testosterone can go right into the body. It's a steroid. Estrogen patches, right? These molecules can go straight through the skin into the bloodstream. Why? Because they are lipid soluble. They can make it through our skin. They can make it into our blood vessels. And if a woman was pregnant, the last thing she'd want to do is have a lot of steroids, and especially sex hormones, going into her body because the baby would also be exposed to them. And if a woman was swimming in testosterone, and it happens, and she has a little girl fetus, little girl might be born with a penis or some weird genitalia because the baby girl had been exposed to too much testosterone, right? Like molecules. So you just don't want to mess with that, right? Let's not go there. So we see that lipid molecules can come straight through the bloodstream, straight into the skin and through the cell membranes. Compared to these little green guys, non-lipid soluble, aka polar, aka hydrophilic, right? They're floating out there in the watery area outside the cell, and they're being rejected by the membrane, right? There's a arrow. Whoop, you're not coming in. You're rejected. Why? They cannot get through the membrane, right? They're being rejected by the membrane. Why? Because these molecules are hydrophilic or polar, and they can't dissolve and go through the membrane. Yes? That's a good question. Why wouldn't they attach? And there might be some of that, but they can't go through. Okay, so the question was, if these are negatively charged heads, why wouldn't some of these molecules be attracted to it, right? Some positively charged molecules might be attracted toward the membrane, and yes, that would be going on, but they wouldn't just stick there. There wouldn't be enough affinity for that. Okay, but good observation. Now, look at these red guys. They're labeled as being some other non-lipid soluble molecule, but it says to us that they're very specific. They are a certain non-lipid molecule, and it turns out that these molecules also would not be able to get to the membrane except they have a helper. Their helper is this membrane protein channel. And because of this red protein channel, these molecules are welcomed into the cell. This is an example, you can write it now, and it'll be on the next slide. This is an example of facilitated, I'm just going to put FAC, diffusion. Okay? This red story right here. Facilitated diffusion is a type of movement where there's a helper, and the helper is this red membrane channel protein. It still, however, is not requiring energy. There's no ATP mentioned. There's no energy involved in any of the processes on this slide. So we see here we've got some issues, right? Some molecules can come in, some molecules are rejected, others can come in, but only when they're welcomed or allowed through by that protein. The other idea here is that it tells us that these are specific molecules. In other words, these green molecules would not be able to get through this pore, right? This is not an open channel. These are specific doors that only allow certain molecules in or out. Okay? So that's kind of getting the idea of lipid and polar, right? Non-polar and hydrophilic and hydrophobic ideas when it comes to the membrane. So these are all passive transport mechanisms. They're all occurring without the use of energy. And in biology, 99% of the time, the energy molecule that we're always talking about is ATP, right? And we talked about how ATP is made by every cell, and it's the energy molecule, and we'll talk about ATP as we need to. So there's different types of passive transport. You're going to be watching these or demonstrating some of these in lab this week. It's about as exciting as watching paint dry, but it's an important idea and concept that we need to all start speaking the same language and thinking about this, not about petri dishes and dyes in the lab, but how is it that molecules move into and out of cells. So there's just good old diffusion or simple diffusion. That's what you'll be doing in lab. Molecules moving from an area of higher concentration to an area of lower concentration. There's another type of diffusion called osmosis. This is a special type only dealing with water, and we'll talk about water and osmosis in lab. Then there's facilitated diffusion. I'll show you a picture of that in a moment, and I had you label that red protein a moment ago as facilitated diffusion. It is still diffusion. It is still passive. Molecules are still moving from higher to lower concentration. But there's a helper. That big red protein channel was a helper. It allowed for it to happen, so it's facilitated diffusion. And then there's bulk transport, and I'll describe that in a moment. That is things like exo and endocytosis. So we'll talk about that in a moment. So here's another picture of facilitated diffusion. I know the D's in the wrong place. So facilitated diffusion. This is how glucose gets into your cells, just as an example. So here, the yellow hexagon molecules are glucose. They're sitting on the outside of the cell. They're in the extracellular fluid, and every cell has to get glucose in for energy. Glucose is too big to just come through the membrane, right? It's got a charge to it, it's got a little of a charge to it. It's too big to just squiggle through the membrane. It is carried into the cell through these purple, in this case, protein channels. There's no energy involved. There's no ATP involved. It's still diffusion. This is facilitated diffusion. So every one of the cells in your body gets glucose into its cell through these facilitated transport mechanisms. And also note, it's still going down the concentration gradient. So molecules are still going from an area of higher concentration on the outside to lower concentration on the inside of the cell. Yes? Yeah, a helper or facilitated. Yep, if you have a facilitator in your life, you have someone who's assisting you, helping you, right, through a process or through an issue. So this is helper, right, helper transport. Okay, again, glucose, key, still no energy, still passive. Now, active transport, and you'll be defining this in your pre-lab for lab 2, is where molecules move but require energy to do so. And when they move, they're going from an area of lower concentration to an area of higher concentration. They're being pushed through the energy to places they wouldn't just go on their own. There's a reason why the body must concentrate or push the molecules into certain areas or certain cells. Whenever there's an active transport mechanism, there's going to be energy necessary to push it. It's sort of like when we talked about enzymes, right? There was an activation energy, something that had to push up the hill that took some energy. That would be like active transport, pushing the ball up the hill. But then once it got to the top, the reaction just goes spontaneously down the hill. That'd be the passive side. So active, pushing something up, passive, things happening on their own. We won't be describing, the only time I'm going to talk about active transport this semester will be a little bit now, and then I'll mention it again at the end of the semester when we get to the kidneys. But there is in almost every one of your cells a very important transporter, you may have heard of it, called the sodium-potassium pump. I'm not going to describe what it does right now, but it requires energy. And without it, we're dead, right? It's just a really, really important mechanism, and that's all I'm going to say right now. When you see an image, when you see a cartoon, and there is active transport going on, you will always see within that image ATP, right? So in this cartoon about active transport, you've got the outside of the cell on the right, you have the inside of the cell on the left. There's a molecule, here's called substrate, there's a molecule that's going to come into the membrane, and this particular transporter is almost like one of those big revolving doors at a hotel or at an airport. And the molecule comes in, and then the door is going to turn or shift, allowing the molecule into the cell. And as we see, for this protein to do its little job and to turn and twist, it requires ATP. Okay, so that's all I'm going to say. There it is, sodium-potassium pump again, as our example. The last thing, so we've talked about how molecules can go straight through the membrane. We've talked about how they can be carried by facilitators. We've talked about how some are rejected, and now in active transport. And now we're going to talk about bulk transport. That is, how does the cell bring in large collection of molecules? And there's three different ways, three basic ways that cells bring in large quantities of stuff. And they're all called endocytosis. Endo, stuff coming into the cell, within the cell. You've heard of them, I'm sure. Phagocytosis is one. Phage, P-H-A-G, means to eat, right? So phagocytosis is cellular eating. And we're going to see that this is a way by which the cell can grab large things and bring them in, things that are way too big to ever fit through a little protein channel, right? Big things can be brought in through endocytosis. Phagocytosis. Then there's penocytosis. It's a variation on the theme. This is what I think of as cellular slurping, cellular drinking. We're not bringing in big chunks, but we're kind of slurping. I think of it like slurping through a little straw. And then when I think of penocytosis and straws, I think of pina colada. So whatever, right? Something we're slurping, we're just kind of taking in a flavor from the environment. And I'll just show this to you in a picture. And then there's a special type called receptor-mediated endocytosis, and cartoon coming. We'll try to make the difference in that in a second. So in this first cartoon, the top picture is phagocytosis. So the top is phagocytosis, the bottom is penocytosis. And what you see, here is the outside of the cell, extracellular fluid or interstitial fluid. In the environment of the cell, there's blue and green and red little molecules, these little guitar pick looking things. There are different molecules in the environment. And there's this big chunk of something, this big green dude. Now that big green thing could be a nutrient molecule, like a big chunk of something, or it could be, if this is an immune cell, this could be a white blood cell grabbing onto a bacterium, right? And it's going to gobble up the bacterium as part of your immune system. But regardless, if it's a big chunk of food or a microbe, it doesn't matter. What happens is that the cell membrane reaches out and like bear hugs this object in the environment. Now, some cells are just kind of doing this randomly, occasionally grabbing things. Others are triggered to do this, but regardless, they kind of reach out, big bear hug with the membrane, and encapsulate and circle this structure, this thing. These membranes are like bath bubbles, right? They kind of blip and blop and connect and separate quite easily because they have the same environment. So this membrane, as we'll see when I'm drawing, will come around and connect on the other side. Those membranes will blip or blop, whatever the word it is, right, for the bubbles to come together. And now, what was on the outside of the cell is now contained inside this vacuole. It's confusing. Some books will use the word vacuole, and then they'll turn around and say the word vesicle. Think of them as the same thing. Right? They both start with letter V. Vesicles, vacuoles, I'm not going to distinguish between the two. But they are little bubbles within the cell that contain certain things. So now you've got a little vacuole, and inside of it, it has that molecule that was on the outside, and it is surrounded by the same stuff as the cell membrane. It has phospholipids and proteins. Whatever was in the membrane is now surrounding it as the membrane of this vacuole. So that's phagocytosis. Down below is pinocytosis, and again we have all these little guitar pick molecules out in the environment, and the cell's not going to reach out and grab, but instead it's going to sort of indent itself or invaginate and just kind of take in whatever's out there. It's random, right? So whatever's out in the environment will make its way into this. It'll be closed off, and now whatever was on the outside of the cell is now on the inside. Those could be nutrient molecules. It could be a molecule that the cell's kind of just checking out its environment, looking for certain signals, all sorts of things that this could be. We're not giving any specific examples right now, but we're slurping small molecules from the environment, cellular drinking. Then, this is that third type, receptor-mediated. Phagocytosis brought in big things. Pinocytosis was cellular random slurping of the environment. Receptor-mediated is like pinocytosis, except it occurs more regulated. So, in this particular scenario, there are these blue molecules in the neighborhood. Let this thing update. Okay, in the neighborhood, there's these little blue molecules, right? Little guitar picks, and there are these little slingshot receptors in the membrane. So these represent specific receptors, and these blue molecules bind to those little yellow slingshots. And when enough of those molecules are in the neighborhood, it will cause the invagination of the membrane. So really the purpose of this process was to bring in these blue molecules, right? There was enough of them in the neighborhood, it triggered the intake of them. However, other molecules like these little red guys, just by chance, come in. So it's specific for the blue molecules, but we're going to get a few other random molecules coming in as well. Just a variation on the theme of pinocytosis. Okay, receptor-mediated, it's a little bit more specific. Regardless, we now have those molecules on the inside of the vesicle. What the cell is going to do with them, all kinds of things. Now, this is really happening, right? We're not just making up these cartoons and wondering. We actually have beautiful micrographs now that show this actually occurring. In the upper right, here is something large, and you can see that the cell is beginning to reach out, and it's going to bear hug that molecule and bring it in, right? Phagocytosis. On the bottom is a serial over time images of some sort of receptor-mediated or a pinocytosis. So there are these molecules out in the neighborhood. Here's the double membrane, and you see it beginning to invaginate, and then it really invaginates, and then you see that it completely closes off on the inside. So whatever was on the outside is now bubbled and bundled and brought into the cell. These are all examples of endocytosis, things being brought into the cell. This is the opposite of exocytosis. not to kill this, right? But exocytosis is kind of reverse, and now the cell is getting rid of things, exiting the cell. Question?
So when we see that figure, there's like a screw or something that that one right for that the top one is bringing into the cell.
Yep. The top one is bringing this green molecule, whatever it is, or it could be a bacterium, into the cell. And it might be bringing in some of the other stuff too, but it's targeting that big thing. On the bottom, and you see at the top, it's reaching out and grabbing it. On the bottom, penocytosis, we're not reaching out, but we are invaginating, indenting in, and grabbing things from the environment. Regardless, things are moving in, into the cell. Then opposite of that is exocytosis. This might be the cell getting rid of waste products. Hormones are released from cells to go elsewhere in the body. So imagine now that the cell has made some protein, some hormone, some molecule, and it needs to get those molecules out of the cell into its environment. Same idea. Here's a vesicle. The vesicle is going to go toward the surface of the cell and bleb, that's my fancy word, with the membrane and open up. And now release whatever's in those vesicles would be released into the neighborhood. It's just really the reverse, isn't it? So what I want you to appreciate is that the cell membrane is not this static, never-changing bubble around a cell membrane. It's constantly ebbing and flowing. There are things coming into the cell, taking some of the cell membrane away. There are things being exocytosed, and membrane then joins with the cell membrane. So the cell membrane is constantly changing. And that's what this picture is highlighting. I like it only because it shows the phospholipid membrane in two different colors. You might first think, oh, well, this is the cell membrane, right? Double layer of phospholipids. And here is a vesicle coming toward the surface, carrying in it some yellow-colored molecules. And that vesicle will bleb with the membrane. But look what happens, right? That's number 2. This is number 3. And then now, once it releases its products, what was vesicle now will become part of the cell membrane. Okay, so imagine again, it's ebbing and flowing, blebbing in and out. The cell membrane is not just a static bag around the membrane, around the cell. It is constantly being influenced and changing. And I'm showing this to you in isolation, like this cartoon, like there's one of these little blebs happening on a cell. This could be happening thousands of times a minute, right, all over the cell. So there's lots and lots of activity at the cellular level that we have, I think, a hard time sometimes imagining because it is so complex. And we can only show it as little tiny, bits and pieces to teach the concepts. So that's the cell membrane. And I've spent by far the most time talking about cell membrane. Any questions, things to clarify about mechanisms that molecules get into and out of this hydrophobic, hydrophilic, polar, nonpolar thing? Is that beginning to sink in and make sense? Anything to clarify there? Okay, so the plasma membrane is an organelle, right? It's a functional part of the cell. The rest of this presentation, next 12, 15 minutes, is going to be over describing other organelles in the cell. And we'll go through them one by one and just talk about what they look like, what they're doing. And again, you're just trying to picture them and know what their function is when it comes to mastering this content. You know that the organelles are the building blocks for cells, right? In our hierarchy, it was atoms, molecules, macromolecules, then I squeezed in there organelles, and then cells. All right, so we're going back now to pick up the organelles as we think about the building blocks for cells. And each organelle has a particular job, and without each organelle doing its job, the cell cannot function normally. There are a lot of diseases where there's a problem with a particular organelle. And I'll describe some of those today. Some people have horrible diseases because their mitochondria don't work well, or their lysosomes don't work well, or their peroxisomes don't work well. So we're going to see that organelles are critical to the overall well-functioning of all of your cells in your body. I can break this up in a couple of different ways. I chose to present first the membranous organelles. That is, the organelles which themselves are made of membrane or have membrane around them. I've already described one, right? The cell membrane would be a membranous organelle. Okay, I'm not going to go back to that one. We've done that one. Additional organelles that have membrane with them or associated with them are the endoplasmic reticulum, smooth and rough, right? That was membrane itself. Then the Golgi, also pretty much membrane itself. There are lysosomes and peroxisomes that I'll describe to you. And then mitochondria also have a membrane around them. So we'll start by going through these 5, and then we'll finish up next time, very shortly, a little bit left over, talking about the non-membrane surrounded organelles. And then that'll finish up this first part of Chapter 3, and we'll continue that conversation next Tuesday. Okay? If I were you, just thinking ahead, I don't want you to get behind with things, but I would not be worrying about Chapter 3 homework right now, right? I mean, your focus is going to be on Chapters 1 and 2 and on the first lab stuff, and you really want to come in next week with any and all questions to clarify and then do really, really well on this exam. And if you really get comfortable with all the 1 and 2 material, you could start looking at Chapter 3 things, but that's not going to be your focus, right? This weekend, you're focusing on things we've already talked about. Hopefully enough time is going by that you've had a chance to review them and are making sense of it. I hope you're taking advantage of our walk-in tutors. Anybody walked in there to see Andrew or Janae? Anybody? Okay, a few of you, good. It's a great place to hang out once in a while. It's a great place to go and meet as a small group amongst people of your own lab group. And that room is open 7 in the morning till 9 o'clock at night most days. And you may not feel too much pressure right now, but when we get to the bones and the muscles and other things, that's a room that has examples of all the structures as well.
Is there a list of like times, or is it just always?
The resource room is always open, 7 in the morning to 9 o'clock at night, Monday through Thursday, Friday till about 3. As long as there's a lab going on, the room is available to you. And then the walk-in hours are posted on Blackboard. Okay, the specific walk-in hours. And there's a little bit of variation on that, so be looking for that. One of our tutors has set hours. One of the other tutors is now in the nursing program, and his schedule's being bounced around because of it. So I'll be posting slightly modifying schedules every 2 weeks or so. They'll be posted outside the room. 249 is right across from the hallway. They'll be posted on that easel, but I'll also try very diligently to send that schedule out for you as well, so you know when to expect them to be there. And then there's also one-on-one tutoring, right? If those times don't work, you can always go through the tutoring center to get a one-on-one tutor. And we have about 5 or 6 folks who are available this semester to be tutors for you. So if that's something you want to do, it might be getting a little bit late for exam 1. But if, as you're preparing for exam 1, you say, you know what, I wish I had signed up for a tutor, or I think I might benefit from that in the future, next week would be a great time to establish that so you're all set for exam 2. Okay? Okay, let's take a look. Rough ER, smooth ER. We saw them in the cartoon. These are membranes that are around right outside the nucleus. 2 types, rough and smooth. The rough ER has on it ribosomes. That's why it's called rough. It looks like there's little studs on it. Ribosomes are going to be critical for making proteins. So rough ER, its job is to make proteins. Okay, that's what we're going to talk about, the rough ER. The smooth ER does not have ribosomes. Therefore, it does not make proteins. Instead, the smooth ER is all about making steroids and fatty acids. So the steroids, cholesterol, some of that is made in the smooth ER. Just that cartoon we saw at the very beginning. Again, 2 flavors, 2 types of ER. Rough with the ribosomes, smooth without. The rough is usually closer to the nucleus. The smooth is a little bit further out, okay, away from the nucleus. There's a little note down here. That's why I included this image. It says that the endoplasmic reticulum, makes proteins. The rough ER makes proteins. The smooth ER makes steroids. We got that. Also makes phospholipids, right? All the steroidal kind of molecules. But here's something I want to make note of. The smooth ER is big time involved with detoxifying alcohol and drugs. When you hear about detoxifying alcohol and drugs, what organ comes to mind? The liver, right? That's what we know. The liver detoxifies. Okay, so if I had liver cells, and I could look into a liver cell, what organelle would I find more of than most cells? Smooth ER. right? So a liver cell would have specialized more smooth ER because that's what liver cells do, right? They're going to have to detoxify. They're going to have more smooth ER. If I had a muscle cell, muscles aren't in the business of making lipids or detoxifying. They're more in the business of using up energy. So if I was looking at a muscle cell, I would see more mitochondria, right? More ATP generation. So the idea here, just to get the idea rolling, different cells in the body, they would all have most of these organelles, but some would have more of or less of these organelles based upon that cell's function. A cartoon of it, again, the rough ER is going to be more closely snugging around the nucleus. Again, it's going to be going all the way around in all directions around the sphere of the nucleus. And further out would be smooth ER. Next is the Golgi. The Golgi apparatus, or just the Golgi. It's named after a dude, so capital G is appropriate. It's a proper word. And what the Golgi does is it's going to package whatever was made by the ER. So what was made, the proteins that were made on the rough ER, the lipids and steroids that were made on the SER are going to make their way to the next series of membranes called the Golgi. The Golgi is like the FedEx or the UPS center of the cell. It's going to take those products and specifically package them and send them out in little packets, okay? So that's what it's doing. It's packaging, boxing, and shipping these proteins out into the cell. And pretty obvious to recognize the Golgi. Again, in this artistic view, it's green. It seems to be green in most cartoons. We even use UPS terminology in describing the Golgi. So we have the receiving side and the shipping side. Okay. So that means that the receiving side is closer to the ER, right? And the shipping side is going further out toward the cell membrane. And you see layer after layer after layer, multiple layers. And as the proteins come in, they're going to be rearranged and modified and repackaged and then sent out. Sometimes packaged in little vesicles, sometimes just sent out individually. But that all varies on what's going on in the cell. And I'll show you a couple more images of that. So the Golgi, sorry about that. Here is a colored electron micrograph. This is kind of cool, color enhanced. And so what we're looking at, this purple would be the nucleolus. And then all the tan would be the nucleus, right? And this little double green membrane would be the nuclear membrane or nuclear envelope. You can see that there's little gaps in it. It's not continuous. And that means that this series of membranes that I'm circling right now, that represents some of the ER. And then this flattened set of membranes, very distinctive flattened set, would be Golgi. Okay? And we'd have proteins coming through the ER, packaged by the Golgi, and then sent out into the cell. So that's the direction of this. And keep in mind, all of those membranes, the nuclear membrane, the ER, and the Golgi, they really just are membranes, and they're all connected one to another. They're not floating around disconnected. It's like they're tethered to the nucleus. They're all connected together. Okay, let's kill it, right? We got this. So the tan, ER, smooth or rough, right? And it's receiving, right? These proteins are being made and blebbed off in membranes. Those membranes, just like little bubbles, are being received by the Golgi, going through the multiple layers, and then out on the shipping side. Some of these proteins would be specifically packaged and will become specific organelles, like the lysosome, okay, that I'll talk about next time. But the lysosome is a specific package set of proteins that we'll talk about. Others would become peroxisomes. I'll talk about those next time. Some of these vesicles, let's say they're hormones, these vesicles, we've already seen this, would go to the membrane and be exocytosed. That's what's showing down here, right? They'd be exocytosed. Other of those proteins, maybe some of those proteins actually need to end up in the membrane. right? The proteins that, those channels, those protein channels were made in the cell and were transported to and stuck into the membrane. So you can imagine the complexity here. This is quite the busy UPS center. And then what's not showing is that some of those proteins would be sent out and they would just be floating out in the cytoplasm all by themselves. And some of these proteins would even go back to the nucleus, right, and affect gene expression and DNA and RNA stuff. So there's a lot more going on than this. This is, again, just a simplistic view of the packaging concept of the Golgi. Question?
So then, like, the vesicles that, become part of the membrane, are those made of a phospholipid bilayer?
Absolutely. So all of this, the nuclear envelope, the nuclear envelope, the ER, and the Golgi are all made of those same phospholipid bilayer, right? So it's ebbing and flowing and, and moving back and forth. So not only do you have membranes becoming part, see how this is becoming part of the membrane? You would also have, what's not shown here, other things being endocytosed and membranes coming in. So this stuff is being constantly moved around and replaced. And that's actually where I'm going to stop. So we're not quite done. Two of those secretory vesicles, right, made here, like some of the, there's a lysosome shown here. Sorry. Here's a lysosome shown here. And there's another kind called the peroxisome. That's where we're heading. I'm going to describe to you what the lysosomes and peroxisomes do. That'll pretty much finish up the membrane organelles. We'll finish up 15, 20, 30 minutes at the most on this section, and then we'll be moving on to things like transcription and translation very quickly, talking about what other things cells do. If you're having any concerns with mastering, come see me right now. Let's get this figured out. And otherwise, have a great weekend. Some of you have lab tomorrow, but I won't be seeing you. And I'll see you all next week.